My name is Miguel Lagos, and I teach biochemistry. I don't do research per se in a lab, but I do read a lot of research related to nutrition and biochemistry and health correlations to that.
My name is Megan Solon. I'm a PhD student in the Environmental Science Department. I work with Dr. Ramon Lovato, and I basically use like cell models to study the effects of different environmental contaminants.
Awesome, thank you for introducing yourselves. I'm going to start off with our first presenter today. We have Angie Gonzalez. Actually, we're going to move her towards the end. We have Somali Shah from UNT, and I'm going to ask that you can start your screen, share your screen when you're ready, and you can start.
Just give me one moment. I'll get started. Hello, my name is Molly Shah, and my project studies plant defense and development, specifically the role of a betaine diterpenoids. My research goal. Betaine diterpenoids are naturally occurring metabolites whose biosynthesis and biological functions have been well studied in gymnosperms. They have extensive biological applications. However, very little is known about the biological function of these diterpenoids in angiosperms. Recent studies of betaine diterpenoids suggest that application of these metabolites affects plant development and defense responses. However, whether these betaine diterpenoids are endogenous signaling metabolites in plant development and defense is unknown. So for this reason, this project will manipulate flowering time and defense by altering the accumulation of betaine diterpenoid in plants by expressing 5 genes. 4 from Burkholderia semiflorens for degradation, and 1 gene from Pichia sitchensis to increase accumulation in order to understand how the amount of betaine diterpenoid in the plant affects development and defense. So some background research. Betaine diterpenoids, they're naturally occurring metabolites. They have many industrial, biological, and pharmacological applications. Rosin, which is often used for the bow of a string instrument, it's obtained as a resin from spruce and pine trees, which is rich in these diterpenoids. So within plants, diterpenoids have various roles. They have environmental interactions, resistance to environmental stress, and defense against pathogens. Recent studies have shown that dihydrobitanol promotes flowering time in plants by aiding the plant in transitioning from the vegetative state to reproductive development. Dihydrobitanol has been shown to move through the plant and promote the accumulation of salicylic acid, an important activator of plant defense. So betaine diterpenoid metabolism in bacteria. So DITQ, DITU, and DITA are genes in Burkholderia semiflorens that metabolize the betaine diterpenoids. DITQ and DITU encode for cytochrome P450 and are involved in the degradation of dihydroiodic acid. DITA encodes a RIS-type deoxygenase and is essential for catabolizing betaine diterpenoids. So it can be hypothesized that decreasing the level of betaine diterpenoids will delay flowering time, while increasing them will promote flowering time. CYP720B4 is the other gene that we use. CYP720B4 originates from Pichia sitchensis, also known as Sitka spruce. It aids in the formation of dihydroabiotic acid within the plants. And overexpression of this plant should increase the amount of betaine diterpenoids in plants. So this project aims to prove that overexpression should promote flowering time. There's the relationship between flowering time and leaf numbers, because that is what we study to understand the development. When the plant's growing vegetatively, the apical meristem produces leaves, and when the plant transitions to reproductive development, the apical meristem switches to producing flowers and stops producing leaves. Hence, the number of leaves at the time of flowering when the inflorescence stem appears is used to measure flowering time. So plants that produce more leaves before flowering are considered to be exhibiting a delayed flowering time, and plants which produce fewer leaves before flowering are considered to be exhibiting an earlier flowering time. So how we determined how a betaine diterpenoid affects development, we first inserted the gene that we were testing into, we molecularly cloned it, and then PCR and gel electrophoresis was used to confirm the presence of the gene in the plasmid. And then once the presence of the gene was confirmed and the plasmid was sent for sequencing to confirm that the construct was in good shape, the plasmid was inserted into agrobacterium so the agrobacterium could transform the plant. Then the plant was transformed through a technique called floral dip, where the flower was dipped into the plate containing the agrobacterium. Once the plant had grown and flowered, the seeds were collected and sterilized to grow into new plants. The plants that had been transformed, we conducted DNA extraction and RNA extraction. DNA extraction to confirm the presence of the gene, and RNA extraction to determine whether the genes were expressed. Once both of those were confirmed, then the seeds were collected from the plants where the genes were expressed. They were sterilized and replanted to conduct the flowering experiment. So for the flowering experiment, the seeds were planted in soil and the number of leaves that had grown on the plant at the time of flowering was counted. This experiment helped determine whether the genes affect the development of plant by monitoring their effect on the phenotype of the plant. Our findings. So prior to the flowering experiment, we conducted DNA extraction and RNA extraction. So this is an example of two of the genes, DITQ and DITU. So for the DNA extraction where our results were positive, we conducted RNA extraction on those same plants and to confirm that it was expressed as well. So for the flowering experiment for Arabidopsis thaliana expressing the DIT gene, we counted the number of leaves at the time of flowering, and then we conducted a t-test. If the p-value was less than 0.05, our results were significant. Each graph line represents a different transgenic line of the plant. So DITQ produced significantly lower number of leaves compared to the wild type, while DITU and DIT-A1 produced significantly more leaves than the wild type. And then we also conducted the flowering experiment for CYP720b4. And from this experiment, the only line that produced significant values was CYP6. From this, we can conclude that lines were generated and some of these transgenic lines showed altered flowering time. Since DITQ produced fewer leaves at the time of flowering than the wild type, indicates that the plant is flowering early. Since the plant is flowering early, the product of the enzyme enhances flowering time. DITU and DIT-A1 produced more leaves prior to flowering. This indicates that the flowering time was delayed. The substrate of DITU and DIT-A1 may be involved in delaying the flowering time. And from CYP720b4, CYP6 produced significantly less leaves, or sample 6 produced significantly less leaves at the time of flowering, indicating earlier flowering time. So future research. Further experimentation can be performed on a larger sample size with more repeated results to confirm that the DIT genes in CYP720b4 are affecting the development of the plants. And further research can be done to monitor the level of betaine diterpenoids within the plant after interaction with the genes using gas chromatography and mass spectrometry. GCMS analysis will be able to provide us with a quantitative analysis of whether the genes increase or decrease the betaine diterpenoid content within the plants. And the impact of the expression of these genes on plant defense has yet to be studied, so future experimentation can be executed to understand how the expression of these genes impact plant defenses. Application, plant interaction with endogenous triggers such as a betaine diterpenoid can be used to advance agricultural industry by allowing one to control the start of flowering time, resulting in flower synchronization, which can be useful in mass harvesting because it will allow farmers to harvest multiple fields at a time. And this project allows us to determine which compounds affect certain portions of the plant, resulting in decreased flowering time, and will allow scientists in the future to genetically engineer the plants to make them resistant to pathogens. So by understanding how betaine diterpenoids affect the development and defense of plants, these natural compounds can be used in the future to boost plant immunity to harmful pathogens, reducing the need to use toxic chemicals. Thank you.
Thank you, Somali. I'm going to go ahead and ask the judges and open the floor if they have any questions they'd like to ask at this point in time.
I do have a question. Very nicely done. My only question is, it's actually, I really liked your project, but my question was just like the CYP gene, is that a cytochrome P450 gene or is it different? Okay, it is?
It is. Mm-hmm.
Thank you. I was just wondering, I was like, I wonder if that's a CYP gene. Thank you.
Mm-hmm.
All right. Have you known of any human-based toxicity for consumption of plants that express this natural antibiotic or antipathogen products, and potential concerns about having plants that express this and then be consuming those products?
No, I have not studied that or looked into that.
Thank you.
Mm-hmm.
And do you know which type of pathogens it makes them resistant against?
No, we just use it as a model plant, so we haven't studied specific pathogens.
Okay, thank you.
Mm-hmm.
If anyone else has any questions, feel free to put them in the chat or raise your hand and we'll call on you to speak. I have about 1 more minute for this presentation. In the meantime, our next presenters will be Shreya Nair and Sid Shah from UNT as well. Before we begin, judges, do you need more time to finish writing feedback?
I'm actually taking notes and I plan to fill it out all at the end, if that's okay.
And then Megan, are we good to continue? Awesome. All right, Shreya and Sid, you're welcome to share your screen and go ahead.
Okay, give me 1 moment. Let me know if y'all can see this.
Yeah, you're good.
Okay. There we go. Where did I don't know, can people hear me?
I can hear you. Your
Okay.
presentation, I'm not able to see at the moment.
Yeah, okay, cool. Okay, are we good now?
Mm-hmm.
All right, thank you so much. All right, hi everyone, my name is Autumn. I'm from Rice University, and for the past semester and a half, I've been looking at something called maladaptive social information in Drosophila melanogaster. So what is social information? Well, social information is basically working smarter instead of harder. And what I mean by that is that you can have, oops. Did my video just pause too?
Yeah, your presentation just went off the share.
Oh my goodness, okay. Technical difficulties.
No worries, don't worry. You're welcome to start over and restart time if you have.
Yeah, I can't see myself anymore, actually.
Let me see.
Like my video?
Your video for us, at least, is still working just fine.
Okay, very odd. All right. Well, okay, yes. So basically, I'm sorry, with technical difficulties, social information is working smarter instead of harder. And so let's say that you are a lone wolf and you can use your own resources to, my slides are just not going.
Let me see. Is there any way you'd be able to share it with me and I can see if I can get it up?
Yeah, I can try that. I'm just not sure how I can move.
If you want, you can try your set one more time. Is there like a specific issue that's happening or?
Yeah, it's just not letting me advance the slides how I want to do it. Okay, well, let's try one more time. So yeah, working smart instead of harder. You can be a lone wolf or you can be using the information of others. So basically what I was saying with that is that you can be a sole individual in the wild, meaning you have to use your own resources, you have to use your own food, or you can learn from others, which is what I'm looking at. So basically using your own information, but instead applying that to other situations. So if you see an interaction, you can take that information and integrate it into your own knowledge. And so you can apply social information to a bunch of different things. You can apply it to gathering food, to finding shelter. And of course, what my information is looking at is to finding mates. And so this is how social information works. Basically, you're taking one fly, and there are two flies. So one of them is an eavesdropper, and it is broadcasting a decision or a type of message. So in this case, we're talking about mating. So one fly is broadcasting a decision to mate, and another one is listening to it. And that's how social information works. Basically, one individual is broadcasting a message, another one is eavesdropping in it and like looking on and gathering the information from that decision. So social information is great. You know, you can work smarter instead of harder. You don't have to take so many resources because, as we know, information and resources are very limited. However, social information is not always great. And that's what I'm looking at in my research. So Drosophila melanogaster, which is the common fruit fly, and maladaptive mate choice copying. So not very many people know that flies actually live in social groups. And that's why we're able to look at social information and social learning. So when selecting mates, we're trying to figure out, do flies use social information even when they're getting harmful information? So this is what we're looking at. We're taking two males, and this is the one on the left is a regular wild type male. So that's basically just a very generic fly that you can find in the wild versus this curling wing male. And the curling wing male is your genetic mutant. And so we're trying to find maladaptive, meaning bad, mate choice copying. So we're essentially taking a female fly and we're letting her make a decision to see if a bad choice is made, will the eavesdropper that we talked about before, will she copy that choice? So we're trying to see if the initial fly chooses this maladaptive mate choice, so this inverted curling wing genetic mutant male, which I made in the lab, which has inverted wings that kind of go upwards. And that's actually really bad because he's not able to do the mating dances as well. He's not able to fly around because his wings are literally inverted upward. So he's supposed to be the bad choice. So here's my setup. I'm taking 3 female selected genotypes, 707, 335, and 365. And these are the ones that went through preliminary trials because you do need to sort out for which ones are having better propensity to mate. I'm also using genotype 40, which is my standard wild type male. That was that initial one with the regular flat wings. And of course, the curling wing. And I'd like to point out that these genotypes, these are just arbitrary numbers. So 335 is not super closely related to 365. They're just arbitrary numbers. So what I do is I basically clear my flies about 5 days before trials, which basically means you've got a full vial of flies right here. And basically I empty them out of the vial, so I'm getting virgins. So this is a really important part of the setup, is that I am using virgin flies to do this. And I'm collecting the virgins and then putting them into an arena. And this is my this is my experimental design. Basically, we have phase 1, where we have an arena, and this is a female. These two are females. There's one in a watchtower, and there's one over here just wandering around. And then these ones with the little like darker spots at the end, those are the males. So here's your curly wing male, and here's your regular type male. And this is phase 1. So this female who's allowed to be wandering around, she's actually in the arena for 40 minutes, and she's allowed to make a mate choice. And here's our eavesdropper that we talked about before. She's the one that's watching this interaction. And then we're going to go into phase 2, where this is the same fly. This one and this one are the same fly. And she now has social information. So she's now seeing this decision, this mate choice. And then she's able to make her own choice between a curling wing and a wild type male. These are different flies. So we make her have her own choice. And we're trying to see whether she's going to use that bad social information or not. And so to conclude, maladaptive social information in Drosophila melanogaster actually does exist. And I think that's a really cool finding because we're actually seeing how really bad information can still be copied. Obviously, we need to do more trials to demonstrate this maladaptive social information and altering the arena design, because sometimes it's kind of hard to see the flies and sometimes they just don't want to mate. There are a lot of trials that we've done where we need to edit the experimental design, because if she's not getting the first phase, if she's not getting the social information, the second choice female is actually not allowed to have any social information because she's never seen anything. So altering the arena design, and we're trying to see, does this change if we don't use virgin males? Because sometimes in flies with more mating experience is actually allowed to more successful in mating. So obviously if you have more experience with it, with the mating dance, and you have different pheromones, you're more likely to be successful in a mate choice copy. So we're trying to see if this phenomenon will change if we don't use virgins. And so the significance, why are we doing this? Well, we're trying to figure out what happens when social information is negative, and how does that impact the evolution of the specimen over time. So as we're moving forward in ecology and evolutionary biology, if you're consistently making bad mate choices, we're trying to figure out how that changes not only your genetics, but your offspring over time. And of course, if it's copied repeatedly, this bad social information, and it's taken through generations of time, how is that going to reflect your gene through evolution? So are you going to start having curly wings because you've mated with so many curly winged males? And if so, how does that impact the decisions of your next offspring? And of course, since I am a neuroscience major here at Rice, I want to see the genetic variation of behavior. So how come 707 is less likely to maladaptive mate choice copy than 335? So understanding how that genetic variation happens over time is something that I've looked at quite a bit. And I'd actually like to do some more statistical analysis, because these are still preliminary results, about what the different genotypes and how their different neural connections change for different cognitive decisions is something I've been looking at quite a bit. And of course, to answer our actual question of, if social information is so good, everyone talks about how great it is in papers, what happens when it's bad? What happens when you're consistently seeing a decision that's bad, and then you start making that bad decision because you wanted to save your own resources? Is that just too much of a risk? You know, if you had just made your own decision and used your own resources and trusted your gut, would you have been mating with this curly winged male? And that's something that we've been looking at over the past few weeks. So just to acknowledge my mentors, thank you so much to Marina. She's a third-year PhD student here at Rice. She's been an amazing mentor. Dr. Saltz, who is the leader of my lab. She's provided all these great resources and mentorship. And finally, gratitude to all the members of the Saltz lab. Thank you guys so much. Here are my references. I'll now take questions, and I'm so sorry for all the technical difficulties.
No worries. Thank you so much, Autumn. We really appreciate your patience through the technical difficulties. I'm going to open the floor up to the judges first to see if they have any questions.
Awesome. Thank you so much. I'm sorry for all the back and forth.
It's fine. This brings me back to my college years too, when I was working with Drosophila.
Oh, really?
Awesome. Yeah, there was a little experiment about genetic mutations. And,
yeah, that is.
But I had a question. When you talk about virgins, you want the female and the male both to be virgins, or do you just want the female to be virgin and have a male with more experience in the dance and the courting process?
Yeah, that's a really great question. So we've always had virgin females. We've always wanted to collect virgins to make sure they don't have any previous social information. But the males, we have decided to look at a little bit varying whether they are virgins or not, because sometimes they don't want to mate because they have no mating experience. And so when they're just put into this arena for 40 minutes and they're like, oh, like I've never even seen a female fly before, because we've separated them and isolated them for 4 days so they will age up. That is a bit of a caveat that we found. And so we have been testing sometimes, okay, what if we put a experienced male in the arena? We've actually seen that there's more likely to mate. So that's what we've been looking at for the past few weeks. So females are always virgins, males, we've been trying to phase them out to see what happens when they aren't virgins.
And another quick question. I know that you're in initial stages, and you mentioned that you did see preference or a selection, that it was actually bad, so repetition of bad behavior. Do you have any like percentage wise of how many times this happened, this repetition of bad selection?
Yeah, awesome. Awesome question. Yes, so it happens about half and half. Sometimes we're able to see the maladaptive choice, but it also takes a bit, because not only does the first female have to make a mating decision, but the eavesdropping female actually also has to make a decision. So we've seen about half and half. But of course, as we do more trials and we try and amp up that the experimental sample and how many flies we're getting, hopefully we'll see a lot more. But as of right now, it's about half and half.
I think you did a great job, and I think that you handled your technical difficulties with absolute grace. So no
Thank you so
worries
much.
there. So I guess like my question is more of, I don't know how to explain this. I'm not very knowledgeable with this type of research, but is there like, what type of, do you get quantitative data out of this type of research, or is it strictly qualitative and you just like describe things? Do you know what I'm saying? I guess I'm just more used to seeing like quantitative data. So like,
Of course. Yeah, no, I totally understand. Basically, we're still in our preliminary stages, but we will run statistical analysis once we have more results.
okay.
We do actually, so this is behavioral ecology. So we're actually describing behavior more often, as you were mentioning of, do you just describe it? It's usually like, it's like a Boolean of yes or no. Is there social information? Yes or no, yes or no. But yeah, we actually will run some statistical analysis to see some qualitative numbers. But as of right now, I'm kind of just describing my results. And obviously being in behavior, a lot of it does have to do with pure observation. But yeah, I would really like to explore what you were mentioning about doing a more sophisticated type of numerical analysis. I think that'd be really interesting to do in the future.
Totally understand presenting preliminary data. So no worries there. I was just curious because I
Yeah,
was
of course. Thank you so much for your question.
no problem.
All right, I just wanted to check if there's anyone else who had any questions. Feel free to chat or either go ahead and ask your question right now. It looks like we won't be able to have Angie Gonzalez present. So this will be the last presentation we have. And for all of the presenters, thank you so much. And y'all did a wonderful job. This is the conclusion of the Biology 1 division. And in a minute, we're going to be taking a picture of everyone in it. So go ahead and turn on your camera if you'd like to be in on it. And you're welcome to either do like your school's hand sign or just smile, whatever works best for you.
Yeah. Oh, well, my screensaver is not letting me. Oh, there we go.
All right, I'm gonna switch to gallery real quick, and then we'll do our picture in 3, 2, 1. Awesome. And then just as a closing note, I'm going to ask that the judges please stay on the Zoom call so they can review comments and deliberate, and the awards will be announced after the keynote speech today, and feedback will be emailed after the conference is over. Our next event is going to be the Graduate School Fair. So if you are interested, you're welcome to leave the Zoom call. That will be starting at 11:15 if I'm not wrong. So you have quite a bit of time for your screen break. And you can refer to the program to find the link to the fair. So yeah, thank you so much, y'all.
Thank you so much for hosting.
Thank you. Thank you for sharing your research.
Thank you.
All right, I'm going to stop the recording for now.